That's what glycolysis does, right there. So you kind of lyse the glucose and each of these things. And I haven't drawn all the other stuff that's added onto that. These are all lots of other things with oxygens and hydrogens, whatever. But each of these 3 carbon backbone molecules are called pyruvate. We'll go into a lot more detail on that. But by glycolysis, it by itself generates, well, it needs 2 ATPs, so it needs 2 ATPs, and it generates 4 ATPs. So net, on a net basis, it generates 2. But each of these 3 carbon backbone molecules are called pyruvate. We'll go into a lot more detail on that. But by glycolysis, it by itself generates, well, it needs 2 ATPs, so it needs 2 ATPs, and it generates 4 ATPs. So net, on a net basis, it generates 2. It generates 2 net ATPs. So that's the first stage. And this can occur completely in the absence of oxygen. I'll do a whole video on glycolysis in the future. Then these byproducts, they get re-engineered a little bit, and then they enter into what's called the Krebs cycle. They enter what's called the Krebs cycle, which generates another 2 ATP. And then, and this is kind of the interesting point, there's another process you can kind of say happens after the Krebs cycle, but we're in a cell and everything's bumping into everything all the time. But it's normally kind of viewed to be after glycolysis in the Krebs cycle. This requires oxygen. So to be clear, glycolysis, this first step, no oxygen required, or doesn't need oxygen. Oxygen can occur with oxygen or without it. Oxygen not needed. Or you could say this is called an anaerobic process. This is the anaerobic part of the respiration. We'll write that down too. Anaerobic. Maybe I'll write it down here. Glycolysis, since it doesn't need oxygen, we could say it's anaerobic. You might be familiar with the idea of aerobic exercise. The whole idea of aerobic exercise is to make you breathe hard because you need a lot of oxygen to do aerobic exercises. So anaerobic means you don't need oxygen. Aerobic means it needs oxygen. Anaerobic means the opposite. You don't need oxygen. So glycolysis, anaerobic, and it produces 2 ATPs net. And then you go to the Krebs cycle. There's a little bit of setup involved here, and we'll do the detail of that in the future. And then you move over to the Krebs cycle, which is aerobic. It is aerobic. It requires oxygen to be around. And then this produces 2 ATPs. And then this is the part that, frankly, when I first learned, it confused me a lot, but I'll just write it in order the way it's traditionally wrote. Then you have something called, I'm using the same colors too much, you have something called the electron transport chain. And this part gets credit for producing the bulk of the ATPs, 34 ATPs. And this is also aerobic. It requires oxygen. So you can see, if you had no oxygen, if the cells weren't getting enough oxygen, you can wrote. Then you have something called, I'm using the same colors too much, you have something called the electron transport chain. And this part gets credit for producing the bulk of the ATPs, 34 ATPs. And this is also aerobic. It requires oxygen. So you can see, if you had no oxygen, if the cells weren't getting enough oxygen, you can produce a little bit of energy, but if you don't, but it's nowhere near as much as you can produce once you have the oxygen. And actually, when you start running out of oxygen, this can't proceed forward. So what happens is some of these byproducts of glycolysis, instead of going into the Krebs cycle of the electron transport chain where they need oxygen, instead they go through a side process called fermentation. For some organisms, this process of fermentation takes your byproducts of glycolysis and literally produces alcohol. That's where alcohol comes from. That's called alcohol fermentation. And we, as human beings, I guess fortunately or unfortunately, our muscles do not directly produce alcohol. They produce lactic acid. So we do lactic acid fermentation. Lactic acid. That's humans and probably other mammals, humans. But other things like yeast will do alcohol fermentation. So this is when you don't have oxygen. It's actually this lactic acid that if I were to sprint really hard and not be able to get enough oxygen, then my muscles start to ache because this lactic acid starts to build up. But that's just a side thing. If we have oxygen, we can move to the Krebs cycle, get our 2 ATPs, and then go into the electron transport chain and produce 34 ATPs, which is really the bulk of what happens in respiration. Now, I kind of said this as an aside, that, to some degree this isn't fair, because while these guys are operating, they're also producing these other molecules, or they're not producing them entirely, but what they're doing is they're taking, I know it gets complicated here, but I think over the course of the next few videos we'll get an intuition for it. In these 2 parts of the reaction, glycolysis and the Krebs cycle, we're constantly taking NAD, or NAD+, and we're adding hydrogens to it to form NADH. This actually happens for 1 molecule of glucose, this happens to 10 NADs, or 10 NAD pluses to become NADHs. And those are actually what drive the electron transport chain. And I'll talk a lot more about it, kind of how that happens, and why is energy being derived, and how is this an oxidative reaction, and all of that, what's getting oxidized, what's being produced. But I just wanted to give due credit. These guys aren't just producing 2 ATPs in each of these stages. They're also producing, actually, combined 10 NADHs, which are then used, which each produce 3 ATPs in an ideal situation in the electron transport chain. And they're also doing it with this other molecule, FAD, which is very similar, but they're producing FADH. Now, I know all of this is very complicated. I'll make videos on this in the future, but the important thing to remember is cellular respiration, all it is taking glucose and kind of repackaging the energy in glucose and repackaging it in the form of, the textbooks will tell you, 38 ATPs. If you take the exam, that's a good number, right? It tends to, in reality, be a smaller number. It's also going to produce heat. Actually, most of it is going to be heat. But 38 ATPs, and it does it through 3 stages. The first stage is glycolysis, where you're just literally splitting the glucose into 2. You're generating ETL, but this is kind of the important thing, is that it was lysed, it was cut in half, and this is what happened in glycolysis. And this happened in the absence of oxygen, or not necessarily. It can happen within the presence or in the absence of oxygen. It doesn't need oxygen. And we got a net payoff of 2 ATPs net. And I always say the net there because remember, it used 2 ATPs at kind of that investment stage, and then it generated 4. So on a net basis, it generated 4, used 2, and gave us 2 ATPs. And it also produced 2 NADHs. 2 NADHs. That's what we got out of glycolysis. And just so you can visualize this a little bit better, let me draw a cell right here. I'll draw down here. Let's say I have a cell that's its outer membrane, maybe it's a nucleus, we're dealing with a eukaryotic cell. Communicate, DNA is formed in form, all spread around like that. And then you have some mitochondria, and there's a reason why people call it the power centers of the cell. We'll do that in a second. So mitochondria have an outer membrane and inner membrane, just like that. I'll do more detail on the structure of a mitochondria, maybe later in this video, or maybe I'll do a whole video on them. That's another mitochondria right there. And then all of this fluidic space out here that's between the organelles, or the organelles you can view as parts of the cell that are, they do specific things. They do specific things, kind of like organs do specific things within our own bodies. So between all of the organelles, you have just this fluidic space. This is just the fluid of the cell, and that's called the cytoplasm. Cytoplasm. And that's where glycolysis occurs. So glycolysis occurs in the cytoplasm. Glycolysis. Now, we all know, in the overview video, we know what the next step is. It's the Krebs cycle, or the citric acid cycle, and that actually takes place in the inner membrane, or the inner space of these mitochondria. Let me draw a little bit bigger. Let me draw mitochondria here. So if this is a mitochondria, it has an outer membrane, it has an inner membrane. If I have just one inner membrane, we call it a crista. If we have many, we call it cristae. Oxidation. And essentially what it does is, it cleaves one of these carbons off of the pyruvate, and so you end up with a 2-carbon compound. You don't have just 2 carbons, but its backbone of carbons is just 2 carbons, called acetyl-CoA. Acetyl-CoA. And if these names are confusing because what's acetyl coenzyme A, these are very bizarre, you can do a web search on them, but I'm just going to use the words right now, so we'll just think simple and look at the big picture. So it generates acetyl-CoA, which is this 2-carbon compound, and it also reduces some NAD plus to NADH. And this process right here is often given credit, or the Krebs cycle, the citric acid cycle gets credit for this step, but it's really a preparation step for the Krebs cycle. Now, once you have this 2-carbon chain of acetyl-CoA right here, you are ready to jump into the Krebs cycle. Long talked about Krebs cycle. You'll see in a second why it's called the cycle. The acetyl-CoA, and all of this is catalyzed by enzymes. And enzymes are just proteins that bring together the constituent things that need to react in the right way so that they do react. So catalyzed by enzymes, acetyl-CoA merges with some oxaloacetic acid, a very fancy word. This is a 4-carbon molecule. This is a 4-carbon molecule right here. These 2-CoA merges with some oxaloacetic acid, very fancy word. This is a 4-carbon molecule. This is a 4-carbon molecule right here. These 2 guys are kind of reacted together, or merged together, depending on how you want to do it. Or like that. All catalyzed by enzymes. It's important, some tests will say, is this an enzyme catalyzed reaction? Yes, everything in the Krebs cycle is an enzyme catalyzed reaction, and they form citrate, or citric acid. Citric acid, which is the same stuff in your lemonade or your orange juice. And this is a 6-carbon molecule, which makes sense. You have a 2-carbon and a 4-carbon. You get a 6-carbon molecule. And then the citric acid is then oxidized over a bunch of steps. And now this is a huge simplification here, but it's just oxidized over a bunch of steps to get the carbons are cleaved off. Both of them get a 6-carbon molecule. And then the citric acid is then oxidized over a bunch of steps. And now this is a huge simplification here, but it's just oxidized over a bunch of steps to get the carbons are cleaved off. Both 2 carbons are cleaved off of it to get back to oxaloacetic acid. And you might be saying, hey, when these carbons are cleaved off, like, let's say this carbon is cleaved off, what happens to it? It becomes CO2. It gets put onto some oxygen and leaves the system. So this is where the oxygen, or the carbons, or the carbon dioxide actually gets formed. And similarly, when these carbons get cleaved off, it forms CO2. And actually, for every molecule of glucose, you have 6 carbons when you do this whole process once, you're generating 3 molecules of carbon dioxide. You're going to do it twice. You're going to have 6 carbon dioxides produced, which accounts for all of the carbons, right? You get rid of 3 carbons for every turn of this. Well, 2 for every turn, but really for the steps after glycolysis, you get rid of this whole oxygen group, which addicted by that. All catalyzed by enzymes. It's important. Some tests say, is this an enzyme catalyzed reaction? Yes. Everything in the Krebs cycle is an enzyme catalyzed reaction, and they form citrate or citric acid. Citric acid, which is the same stuff in your lemonade or your orange juice. And this is a 6 carbon molecule, which makes sense. You have a 2 carbon and a 4 carbon. You get a 6 carbon molecule. And then the citric acid is then oxidized over a bunch of steps. And this is a huge simplification here, but it's just oxidized over a bunch of steps to get the carbons are cleaved off. Both 2 carbons are cleaved off of it to get back to oxaloacetic acid. And you might be saying, hey, when these carbons are cleaved off, like, let's say this carbon is cleaved off, what happens to it? It becomes CO2. It gets put onto some oxygen and it leaves the system. So this is where the oxygen or the carbons or the carbon dioxide actually gets formed. And similarly, when these carbons get cleaved off, it forms CO2. And actually, for every molecule of glucose, you have 6 carbons. When you do this whole process once, you're generating 3 molecules of carbon dioxide. You're going to do it twice. You're going to have 6 carbon dioxides produced, which accounts for all of the carbons, right? You get rid of 3 carbons for every turn of this. Well, 2 for every turn, but really for the steps after glycolysis, you get rid of 3 carbons. You're going to do it for each of the pyruvates. You're going to get rid of all 6 carbons, which will have to exhale eventually. But this cycle, it doesn't just generate carbons. The whole idea is to generate NADHs and FADH2s and ATPs. So, right here. And this is a huge simplification. I'll show you the detailed picture in a second. We'll reduce some NAD into NADH. These are in separate steps. There's intermediate compounds. I'll show you those in a second. NAD plus, another NAD plus molecule will be reduced to NADH. It will produce some ATP will turn to some ADP will turn to ATP. ADP turning to ATP. Maybe we have some, not maybe, this is what happens. Some FAD gets, let me write it this way. Some FAD gets oxidized into FADH2. And the whole reason why we even pay attention to these, you might think, hey, cellular respiration is all about ATP. Why do we even pay attention to these NADHs and these FADH2s that get produced as part of the process? The reason why we care is that these are the inputs into the electron transport chain. These get oxidized or they lose their hydrogen in the electron transport chain, and that's where the bulk of the ATP is actually produced. And then maybe we'll have another NAD get reduced or gain in hydrogen. Right? Reduction is gaining an electron or gaining a hydrogen whose electron you can hog. NADH. And then we end up back at oxaloacetic acid, and we can perform the whole citric acid cycle over again. So now that we've written it all out, let's account for what we have. So depending on, let me draw some dividing lines so we know what's what. So this right here, everything to the left of that line right there is glycolysis. Glycolysis, we learned that already. And then most, especially introductory textbooks, will give the Krebs cycle credit for this pyruvate oxidation, but that's really a preparatory stage. The Krebs cycle is really formally this part where you start with acetyl-CoA, you merge it with oxaloacetic acid, and then you go and you form citric acid, and which essentially gets oxidized and produces all of these things that will lead to either directly produce ATP or you do it indirectly in the electron transport chain. But let's account for everything that we have. So far. We already accounted for the glycolysis right there, 2 net ATPs, 2 NADHs. Now in the citric acid cycle or the Krebs cycle, well, first we have our pyruvate oxidation that produces 1 NADH. But remember, if we want to say what are we producing for every glucose, this is what we produce for each of the pyruvates, right? This was from 1 NADH from just this pyruvate. But the glycolysis produced 2 pyruvates. So everything after this, we're going to multiply by 2 for every molecule of glucose. So I'll say so the pyruvate oxidation times 2 means that we got 2 NADHs right there. And then when we look at this side, with the formal Krebs cycle, what do we get? We have how many NADHs? 1, 2, 3 NADHs. So 3 NADHs times 2. We're going to perform this cycle for each of the pyruvates produced from glycolysis. So that gives us 6 NADHs. We have 1 ATP per turn of the cycle. It's going to happen twice, once for each pyruvic acid. So we get 2 ATPs. And then we have 1 FADH2. We're going to do this cycle twice. This is per cycle, so times 2. We have 2 FADHs. Now, sometimes in a lot of books, these two NADHs, or per turn of the Krebs cycle, or per pyruvate, this one NADH, they'll give credit to the Krebs cycle for that. So sometimes instead of having this intermediate step, they'll just write 4 NADHs right here, and they're going to do it twice, once for each pyruvate. So they'll say 8 NADHs get produced from the Krebs cycle. But the reality is 6 from the Krebs cycle, 2 from the preparatory stage. Now, the interesting thing is we can now count whether we get to the 38 ATPs promised by cellular respiration. We've directly already produced for every molecule of glucose 2 ATPs and then 2 more ATPs. So we have 4 ATPs. 4 ATPs. How many NADHs do we have? 2, 4, and then 4 plus 6 is 10. We have 10 NADHs. And then we have 2 FADHs, 2 FADH2s. I think in the first video on cellular respiration I said FADH. It should be FADH2 just to be particular about things. And these, so you might say, hey, where's our 38 ATPs? We only have 4 ATPs right now. But these are actually the inputs in the electron transport chain. These molecules right here get oxidized in the electron transport chain. Every NADH in the electron transport chain produces 3 ATPs. So these 10 NADHs are going to produce 30 ATPs in the electron transport chain. And each FADH2, when it gets oxidized, it gets turned back into FAD in the electron transport chain, will produce 2 ATPs. So 2 of them are going to produce 4 ATPs in the electron transport chain. So we now see we get 4 from just what we've done so far, glycolysis, the preparatory stage, and the Krebs or citric acid cycle. And then eventually these outputs from glycolysis and the citric acid cycle, when they get into the electron transport chain, are going to produce another 34. So 34 plus 4, it does get us to the promised 38 ATP that you would expect in a super efficient cell. Kind of theoretical maximum in most cells. They really don't get quite there, but this is a good number to know if you're taking the AP Bio test or most introductory biology course. There's one other point I want to make here. Everything we've talked about so far, this is carbohydrate metabolism, or sugar metabolism, we can call it. We're breaking down sugars to produce ATP. Glucose was our starting point. But animals, including us, we can catabolize other things. We can catabolize proteins. We can catabolize fats. And if you have any fat on your body, you have energy. In theory, your body should be able to take that fat and you should be able to do things with that. You should be able to generate ATP. And the interesting thing, the reason why I bring it up here is obviously glycolysis is of no use to these things, although fats can be turned into glucose in the liver. But what the interesting thing is that the Krebs cycle is the entry point for these other catabolic mechanisms. Proteins can be broken down into amino acids, which can be broken down into acetyl-CoA. Fats can be turned into glucose, which actually could then go the whole. The general catabolic biology, cellular respiration initially, because there's just so much information, it's hard to process what's important. But I want to just highlight the important steps here, just so you see it's the same thing that we talked about. From glycolysis, you produce 2 pyruvates. That's the pyruvate right there. That actually shows molecular structure. This is the pyruvate oxidation step that I talked about, the preparatory step. And we reduce an NAD plus into NADH. The step that I talked about, the preparatory step. And you see we produce a carbon dioxide and an NAD plus into NADH. Then we're ready to enter the Krebs cycle. The acetyl-CoA and the oxaloacetate, or oxaloacetic acid, they are reacted together to oxaloacetic acid. They are reacted together to create citric acid. We've actually drawn the molecule there. And then the citric acid is oxidized. Krebs cycle. The acetyl-CoA and the oxaloacetate or oxaloacetic acid, they are reacted together to create citric acid. We've actually drawn the molecule there. And then the citric acid is oxidized through the Krebs cycle right there. Each of these steps are facilitated by enzymes, and it gets oxidized, but I want to highlight the interesting parts. Here, we have an NAD get reduced to NADH. We have another NAD get reduced to NADH. And then over here, another NAD get reduced to NADH. So far, if you include the preparatory step, we've had 4 NADHs formed, 3 directly from the Krebs cycle. That's just what I told you. Now we have, in this diagram, they say GDP or GTP gets formed from GDP. The GTP is just guanosine triphosphate. It's another purine that can be a source of energy, but then that later can be used to form an ATP. So this is just the way they happen to draw it, but this is the actual ATP that I drew in the diagram on the top. And then they have this Q group, and I won't go into it, and then it gets reduced over here. It gets those 2 hydrogens, but that essentially ends up reducing the FADH2s. So this is where the FADH2 gets produced. So as promised, we produced for each pyruvate that's inputted, remember, so we're going to do it twice. For each pyruvate, we produced 1, 2, 3, 4 NADHs. We produced 1 ATP and 1 FADH2. That's exactly what we saw. Interesting part. Here we have an NAD get reduced to NADH. We have another NAD get reduced to NADH. And then over here, another NAD get reduced to NADH. So far, if you include the preparatory step, we've had 4 NADHs formed, 3 directly from the Krebs cycle. That's just what I told you. Now we have, in this diagram, they say GDP or GTP gets formed from GDP. The GTP is just guanosine triphosphate. It's another purine that can be a source of energy, but then that later can be used to form an ATP. So this is just the way they happen to draw it, but this is the actual ATP that I drew in the diagram on the top. Then they have this Q group, and I won't go into it, and then it gets reduced over here. It gets those 2 hydrogens, but that essentially ends up reducing the FADH2s. So this is where the FADH2 gets produced. So as promised, we produced for each pyruvate that's inputted, remember, so we're going to do it twice. For each pyruvate, we produced 1, 2, 3, 4 NADHs. We produced 1 ATP and 1 FADH2. That's exactly what we saw up here. I'll see you in the next video. After being done with glycolysis in the Krebs cycle, we're left with 10 NADHs, and 2 FADH2s. And I told you that these are going to be used in the electron transport chain, and they're all sitting in the matrix of our mitochondria. And I said they're going to be used in the electron transport chain in order to actually generate ATP. So that's what I'm going to focus on in this video. The electron transport chain. And just so you know, a lot of this stuff is known, but some of the details are actually current areas of research. People have models, and they're trying to substantiate the models, but things are happening at such a small scale here that people can just look at the evidence, some of it of which is indirect, saying this is probably what's happening. Most of this is very well established, but some of the exact mechanisms of, for example, how exactly some of the proteins work aren't completely understood. So I think it's very important for you to understand that this is kind of at the cutting edge, and you're already there. So the basic idea here that the NADHs, and that's where I'll focus, FADH2 is kind of the same idea, although its electrons are at a slightly lower energy state, so they won't produce quite as many ATPs. Each NADH, maybe I'll write it here, each NADH is going to be, as you'll see, indirectly responsible for the production of 3 ATPs, and each FADH2, in a very efficient cell, in both of these cases, will be indirectly responsible for the production of 2 ATPs. And the reason why this guy produces fewer ATPs is because the electrons that he has going into the electron transport chain are at a slightly lower energy level than the ones from NADH. So in general, I just said indirectly, how does this whole business work? Well, in general, NADH, when it gets oxidized, NADH, remember, oxidation is the losing of electrons or the losing of hydrogens that happen to have electrons. You can write the TAP reaction like this. It's oxidation reaction like this. You'll have some NAD+, which you can then go and use back in the Krebs cycle and in glycolysis. You have some NAD+, you'll have a proton, right? A positive hydrogen ion is just a proton. And then you'll have 2 electrons. This is the oxidation of NADH. Oxidation of NADH. It's losing these two electrons. Oxidation is losing electrons. Oil rig. Oxidation is losing electrons, or you can imagine it losing hydrogens from which it can hog electrons. Either one of those is the case. Now this is really the first step of the electron transport chain. These electrons are transported out of the NADH. Now the last step of the electron transport chain is you have two electrons. You can view it as the same two electrons if you like. Two electrons plus two hydrogen protons. And obviously if you just add these two together, you're just going to have two hydrogen atoms, which is just a proton and electron, plus one oxygen atom. So I can say one half of molecular oxygen. That's the same thing as saying one oxygen atom. And you're going to produce, if I have one oxygen and two complete hydrogens, I'm left with water. I'm left with water. And you can do this. We're adding electrons, or we're gaining electrons to oxygen. Oil rig. Reduction is gaining electrons. So this is the reduction of oxygen to water. This is the oxidation of NADH to NAD+. Now, these electrons that are popping out of, these electrons right here, that are popping out of this NADH, and when they're in NADH, they're in a very high energy state. And what happens over the course of the electron transport chain is that these electrons get transported to a series of, I guess you'd call them transition molecules, but these transition molecules, as the electrons go from one to the other, they go into slightly lower energy states. And I won't even go into the details of these molecules, coenzyme Q, cytochrome C, and then they eventually end up right here, and they're used to reduce your oxygen into water. Now, every time an electron goes from a higher energy state to a lower energy state, that's what it's doing over the course of this electron transport chain, it's releasing energy. So energy is released when you go from a higher state to a lower state. When these electrons were in NADH, they were in a higher state than they are when they bond to coenzyme Q. So they release energy. Then they go to cytochrome C, release energy. Now that energy is used to pump protons across the cristae, across the inner membrane of the mitochondria. This is all very complicated sounding. And this is the cutting edge. So it maybe should sound a little complicated. So let me draw a mitochondria. So let me draw a small mitochondria just so you know where we're operating. That's its outer membrane, and then its inner membrane, or its crista, look like that. Looks just like that. And let me zoom in of the membrane. So let's say if I were to zoom in right there. If I were to zoom that out, that box would look like this. You have your crista here, and I'm going to draw it thick. Right, so I'm zooming in. This green line right here, I'm going to draw it really thick. Color it in with green, just like that. And then you have your outer membrane. This outer membrane, I can do it up here. I'll just color it in. You see the outside of the outer membrane. Right here, this space right here, this is the outer compartment. And then we learned in the last video, this space right here is the matrix. This is the matrix. This is where our Krebs cycle occurred, and where a lot of our NADH, or really all of our NADH, is sitting. So what happens is every time NADH gets oxidized to NAD+, or each of as the electrons keep transferring from one molecule to another, it's occurring in these big protein complexes. I'm not going to read details of this. So each of these protein complexes span. So that's the protein complex where this first oxidation reaction is occurring and releasing energy. And then let's say there's another protein complex here where the second oxidation reaction is occurring and releasing energy. And these proteins are able to use that energy to essentially pump, this might all seem very complicated, to essentially pump hydrogens into the outer membrane. It actually pumps hydrogen protons, let me be very clear, hydrogen protons into the outer membrane. And every one of these reactions pump out a certain number of hydrogen protons. So by the end of the electron transport chain, or we just followed one set of electrons, by the time that they've gone from their high energy state in NADH to their lower energy state in water, by the time they've done that, they've supplied the energy to these protein complexes that span our cristae to pump hydrogen from the matrix into the outer membrane. So really the only byproduct of the oxidation of NADH into eventually water, or the oxidation of NADH and the reduction of oxygen into water, isn't ATPs yet. It's just this gradient where we have a lot higher hydrogen proton concentration in the outer compartment than we do in the matrix. Or you can say that the outer compartment becomes a lot more acidic. Remember, acidity is just hydrogen proton concentration, the concentration of hydrogen protons. So the byproduct of this, all of this energy is used to really just pump these protons into the outer membrane. So you have two things. The outer membrane becomes more acidic than the matrix inside. We can call that basic. And then obviously these are all positively charged particles. So there's actually an electric gradient, or electric potential between the outer membrane and the inner membrane. This becomes slightly negative, that becomes slightly positive. These guys wouldn't naturally do this on their own. If this is already acidic and it's already positive, left to its own devices, these more protons wouldn't be entering. And the energy to do that is supplied by electrons going from high energy state in NADH to going to a lower energy state eventually on the oxygen in the water. That's what's happening. But essentially all that's happening is protons being pumped from the matrix into the outer compartment. Now, once that gradient forms, these guys want to get back in. These guys want to get back in to the matrix, and that is where the ATPs are born. So there's a protein that also spans this. Let me draw remember, this is all this inner membrane right here. Let me just draw it a little bigger right here. So that's our inner membrane, our cristae right there. There's a special protein called, and I'll show you actually a better diagram of what it looks like in a second, called ATP synthase. ATP synthase. And what happens is, remember, because of the electron transport chain, we have all of these hydrogen ions up here, all of these protons really, well they are protons, that really want to get back into the matrix down here. They really want to get back down to the matrix, but they can't. This cristae is impermeable to them, so they have to find a special way to get through. They were able to go the reverse direction through these special protein complexes. Now they're going to go back into the matrix through ATP synthase. So they're going to go back, but something interesting happens. And this is really an area of current research where people are, they think they know how it works, but they're not sure, because you can't just take these proteins apart and operate them, or just watch them operate like you can a regular mechanical engine. These are ultra small, and they have to be in a living system, and they have to have the right conditions, and you can't even, it's hard to see hydrogen protons. These are ultra small things that are, pretty much you can't see them. But what happens, the current model is that as these enter, as these go through my ATP synthase, there's actually an axle. So you can kind of view this as a housing, and then there's an axle. And this is all just a big protein. There's an axle, and then there's another part of the synthase down here. So you can imagine, this is kind of mind-blowing. Something this fancy is occurring on the membranes of pretty much all living systems cells. It's not just eukaryotes, but prokaryotes do this. They don't do it in mitochondria, they do it in their cellular membrane, but it's a pretty neat thing. And what happens is, as these go through, you can kind of imagine this water flowing through a turbine. It mechanically causes this structure in the middle to spin, to actually spin. This is the current thing like that. Randomly, but it's right here. And so as this turns, the outer housing, because of just the proteins bumping against each other, just enough protons to put into our H2 level. Sometimes electrons might somehow jump the gun or jump some steps, so some of the energy gets lost. So you don't always have a completely efficient system. And just so you believe that this is actually occurring on our membrane, there's actual visual depictions of these proteins. This is the actual protein structure of ATP synthase. Relatively stationary spot. Some stick or not a windmill is, or maybe the simple, any kind of, if you want, introductory. Electrons are moving from the NADHs and the FADH2s to eventually show up and reduce the oxygen. And as they do that, they're releasing energy as they go from one. Molecule to another. They're going to lower energy states. That energy is used to pump hydrogen protons into the outer compartment of the mitochondria. And then that gradient, those hydrogen protons want to get back into the matrix of the mitochondria. So as they go back in, that drives this ATP synthase engine, which actually produces the ATP. So just like we said in the past, when you have 10 of these, on average, on average, each NADH is going to produce 3 ATPs, not directly. It produces enough of a gradient of hydrogen protons to produce 3 ATPs in the ATP synthase, and each FADH2, on average, produces enough of a hydrogen gradient to produce 2 ATPs. So if we come in with 10 NADH, they're going to produce, in this ideal world, 30 ATP. And then our 2 FADHs, H2s, are going to produce 4 ATP. And then if you remember, from glycolysis, we had 2 net ATPs directly produced, and from the Krebs cycle, we get 2 ATPs directly produced. So then you have 4 from glycolysis and Krebs, and that gets us once again to our magic 38 ATP from one molecule of glucose. And now I think you have a pretty good grasp of cellular respiration. I made a slight error in the electron transport chain video, and I wanted to correct it in this one, and it's also an opportunity for me to include a little bit of terminology that I forgot to include in that video. So when I described the electron transport chain, you remember it's just you have some high energy electrons in NADH, and they get transferred from one molecule to another, and as they get transferred, they go into lower energy states, and they release energy. And then the final electron acceptor was oxygen. Oxygen got reduced right here, but if you do was, I need 2 hydrogens. I have 2 hydrogens on the right-hand side of the water. I need 2 hydrogens on the left-hand side. So there should be 2 right there. So that was what I would consider to be a minor mistake in the last video. But this also gives me a chance to introduce you to some more terminology. So this whole process, we know that this is called oxidation, right? When NADH loses a hydrogen, remember oxidation is log that hydrogen's electrons. So this whole process of the electron transport chain is one molecule after another getting oxidized until you have a final electron acceptor. In water. So this is obviously, you could call this oxidation, just very generally. And then the second part of the electron transport chain, or maybe we should even call this part of the electron transport chain, the process where the ATP is actually formed, the adding of a phosphate group into another molecule is called phosphorylation. Phosphorylation. So the whole process of creating ATP through the electron transport chain. Remember, the electron transport chain releases energy that creates this hydrogen gradient, it pumps the hydrogen to the outer compartment, and then that gradient, those hydrogens that want to get the adding of a phosphate group into another molecule is called phosphorylation. Phosphorylation. So the whole process of creating ATP through the electron transport chain. Remember, the electron transport chain releases energy that creates this hydrogen gradient, pumps the hydrogen to the outer compartment, and then that gradient, those hydrogens that want to get back into the matrix, essentially go back through this ATP synthase. Phosphorylation. It's a good word to know. You might see it on some standardized tests or on your exams. It's called this because you have an oxidative part. Each of these molecules get oxidized in the electron transport chain as they lose their hydrogens or as they lose their electrons. That creates a hydrogen gradient. And then that, through chemiosmosis, allows for phosphorylation. So that's another good word to know. The transfer of these hydrogens, these kind of going through this membrane selectively. This membrane, this ATP synthase wouldn't allow just any molecule to go through it. It's allowing these hydrogen protons to go through it. This process right here, this hydrogen going through it, is called chemiosmosis. Another good word to know. So the entire process is called oxidative phosphorylation. They don't happen at the same time. Oxidative generates the energy because the energy to push the hydrogens out, and the phosphorylation happens as the hydrogens experience chemiosmosis and go back in and turn this little axle and then push the ATP or the ADP and the phosphate groups together. And then you can contrast that with substrate phosphorylation, since we're in the mood to introduce you to terminology. Substrate phosphorylation. This is actually what happens when the ATP is produced directly in glycolysis and the Krebs cycle. So this is the glycolysis and the Krebs cycle. And this is where you have an enzyme directly helping to produce the ATP without any type of chemiosmosis or proton gradient. So if you imagine an enzyme, some big protein blurb, and let's say it has the ADP there with its two phosphate groups, and then maybe it has another phosphate group that attaches to some other part of the enzyme. This enzyme facilitates without any kind of chemiosmosis or oxidation. It facilitates probably in conjunction with other energy releasing reactions that maybe are occurring in other parts